Alright guys, alright here we go. The official start of our studying video. I'm uploading the other video right now, the video before you see this, so yeah. I don't think my friend can join. Well, I don't think I can talk to my friend, like call him. But I was gonna call him because, you know, we're in the same class in biology, but see, this is the kind of thing, you never know what's gonna be on the big test in biology. In math, you can kind of see it, because, you know, we review a bunch of stuff in math, but science is, it's just, or biology in general is just weird to learn. We mostly learn about genetics, so, and that was pretty much my final project was about, the 300 points. I'm gonna go over that with you guys, but first I gotta eat something, so if you guys want to eat with me, go get you something to eat. Right now I'm eating dinner. I plan not to eat on camera, but that's just the way it goes sometimes. Drink is Powerade. Sorry for the moment of silence. Ten phrases on the scene there in Mesa. We got some lights going in Sacramento. Units wasting sunlight. Let's check in. You know what I could have done? I could have paused the video and then just eat, and then I can just unpause it where I left off. But you know what? It don't matter because I'm almost done eating, so yeah. Good thing this camera, this webcam doesn't take long to upload. Yeah. I'm watching this new show, Live Rescue. It's basically, if you know what Live PD is, and you know what I'm talking about. It's like firefighters and EMTs responding. I don't really like watching that stuff, but you know, it's on every Monday night, so it's Monday night right now. So yeah. Okay, so sorry for not talking much, but I'm just really hungry sometimes. Probably because, you know, this is what happens when you study, you get hungry. Here's my dessert. I guess I can show you my dessert. Donuts. Woohoo! The little tiny ones, even though there's no hole in it, but still donut. Not the Chinese donuts. Those are good too, but I like this better. You know what I should call it? Donut rolls. Because it looks like a size, it's like a roll, like a bread, like mini rolls. I can call it that, a mini donut roll. Yeah. Okay, before we start, I want to check my channel. Again, this is my backup channel, so if you want to, like, this is only, school-related stuff, and I guess you can say goodnight streams. Oops. If you want to see non-school stuff and more of me, that's my real channel, Eric Super Magnificent, so you can find the name in this channel right here. Yeah, those test streams, I should have deleted them all. I don't know why, because I barely checked this channel. That's probably why. Oh, wow. I forgot I did a video about the problem of one high school class, which is biology, and I had an F. But it went back up to a low C, so if I do good on that 300-point essay, I should be good. I don't see how I can do really bad or just bad, because I put the links of where I got the information of. Yeah, I did a high school update, so yeah, I haven't uploaded a video besides the test streams, since March 23rd, but besides those test streams, oh wait, uh, shoot, not that one. March 6th, so, oops, sorry about that, that was me. So it's been like pretty much exactly 2 months. Oh, thank you for subscribing. Last time I checked, I think I had like 50 subs, so now I have 61? I just gained like 11 subs. For this backup channel, I'm kind of surprised. So thank you guys. I know I don't have a banner. I don't even know what to put on the banner, to be honest. Maybe, I don't know. I've noticed, you know how, the new YouTube layout has changed? You can go to the old school YouTube layout. I like the new one because you can have the dark theme on. Yeah, you know how when you're in the old YouTube layout, the banner's just a bunch of black bricks? But now they don't have it here. Anyway. Yeah, I haven't done a good night stream since, what, 4 months ago? So it's like around Christmas. Oh, wow. I'm checking my old videos. I did studying for the midterm. Why did I do a live stream of that? I think it's because I want to talk to you guys. And, the midterm, it's not as big as this one, so I guess that's probably why. I don't know. Let me check these out real quick. Like, not many people were watching it, so that's another reason why this is not live. Plus, I don't really have time to do it live. Is this on OBS or Google? Oh, this is on OBS. Yeah, those test streams was from OBS, and that's why I want to do a legit good live stream on my main channel using OBS, but for some reason OBS wasn't working that day. So I don't really trust it now. Maybe I can go back to it during the summer, but right now, it's just gonna be not live. So, yeah. And plus, I don't think any of you guys will be watching it, because if you have school like me, then you guys will be probably doing the same thing as me, so, yeah. I forgot what I was doing in this video. What was I doing? Oh, it wasn't OBS, it was the go live button uh below the upload button. What was I doing? I have no idea. Oh, was I just showing my dusty laptop? I have no idea what the point of that is. Yeah, I don't really screen share what I'm studying. I just talk. Sorry guys, I'm gonna turn up. I wanna hear what I'm saying. Three's only 2 questions. This is a big one. This is a weird. She said, I forgot to tell you, she said it's gonna be sort of easy, so hopefully that will help. Unit 2. Yeah, see, when I did these studying videos of the midterm, they gave me, like, papers. But now, that's for, like, every class. So pretty much the midterm is going to be kind of like the finals. Which finger controls the Q key or not? Yeah, that's why these videos were long. So, oh, I was on the phone too. How long would this video be? I don't know. Probably maybe an hour and a half, maybe 2 hours tops. I don't want to make it too long. Because biology, you don't really exactly know what's going to be on the test. So, and if it's just going to be reading, then I don't really have to study much. Maybe, I mean, I'll study enough. Oh, I was gone for like almost an hour, just me pooping. Wow, I'm an idiot. I should have ended the video when I had to poop. Those thumbnails. The reason why I did those thumbnails is because it tells you to smile for the thumbnail or do like a picture or a pose, so that's what I did. That's what that was. And why was the cameras like pixelated, or the video? I don't know why. Because, you know, I don't really watch the stream while I'm talking, while I'm live streaming, so... Yeah. It's a good thing I'm not doing that again. Oh, shoot. I hope I didn't hit anything. What? Oh, shoot. I'm sorry that I'm so close to the camera. I don't know why. Like, I'm not actually that close to the camera. I think it's just how the video is. Sorry about that. Oh. So I did 2 days? And another reason, the camera app, it stopped, like, I think my internet, like, screwed this up. So when you have, like, bad internet during a live stream, this is what happens. Stop! Oh my god. I did this, like, prank that did not work well. Part 3. I was pretending, this is gonna sound really weird. You know how, so this was a live stream back in December, and I look at the chat while I'm talking, and then I want to see if the sound is good, and then I'm pretending I'm talking to myself on the phone while it's a live stream. So basically, there's 2 Erics talking to each other, and like I'm saying something, and then like the live stream is like probably like 10 seconds behind, because you know it's not completely the exact time, but he pretty much says the same thing after one another. And I did that since the beginning until I did that for like over a half an hour? No way. Why did I do this? Okay, I was on the phone with someone else. Jesus Christ. What the heck?
The midterm should be like something different.
I can't believe the last live stream was over 5.5 hours long. Oh, I was gone for a long time. Do I have to poop? Oh, wait, no, I didn't. I have no idea what. I just had my earbuds in. I did not. Maybe I forgot this was a live stream? I have no idea. This is an awesome live stream. Go ahead and watch that 5.5 hour studying for the midterm exams day 2 live stream part 3. It's pretty much for the first 45 minutes just me on the phone, and then the rest is just me being quiet. And then you got like 30 minutes of me being gone, and then I came back. Jeez. Okay, I know we don't have time to play around, but what the freak? Okay, well, that's fun to watch, I guess. And if you're wondering why those the last two good night streams were so long, so I'm guessing 11 hours and 55 minutes is the longest time you can do a live stream just from the camera. That's a long time. I did not sleep the whole 11 hours, 55 minutes. I just left it on because it was at my brother's house. And I don't know why. I just forgot to stop it. I was being stupid, I guess. Okay, I did a high school update last year of the state testing and finals. Okay. Last year I did a live stream of preparing for testing and other stuff. I was talking to Ryan. This was April 21st. See, I didn't do any midterms of 2017 because we didn't really have much. So yeah. All right, all right. Before I start, I gotta put this plate in the dishwasher or the sink. So I'll be back. Don't worry, I'm not gonna take a poop. I already took a poop, so good. All right, I'll be right back.
handpicked by the pickiest car flipping people out there. Real cars are carefully inspected and come with a Carfax. Sweet ride. Thousands of cars priced below market value. Hey, just go to vroom.com, pick your favorite, and they'll deliver it right to you. Congratulations! Is this for real? It's 32 pounds. 168 pounds. 59.4 pounds. Do you feel healthier on WW? Do you worry about keeping it off? There's no stopping. I said after it. It's not even a diet. It's a lifestyle. Weight Watchers is now WW. Join today with the WW Triple Play. Offer ends May 6th. Meet the Ninja Foodie, the pressure cooker that crisps. It's the best of pressure cooking and air frying all in one. So in as little as 30 minutes, it will be crispy on the outside, juicy on the inside, and on your table. The Ninja Foodie, the pressure cooker that crisps. With the incorrect, what you expect from all that. Because none of your crazy as that sounds, this character is incredibly real to me. And Jeff Foxworthy. A lot of people don't know if they're a redneck or not. I don't know what I would ever want to say. I figure out what my feel like, but maybe if I get over it too far. Two legendary comedians, one special night, biography, Jeff Dunham, May 28th. Then Jeff Foxworthy at 10 on A&E. Live Rescue. We have a car rollover this Thursday. I need you to be okay. I know I'm going to get you. Catch a bonus episode. Cable's new #1 nonfiction series. Don't miss a bonus episode of Live Rescue Thursday at 9 on A&E. Now for tonight's Citizen Hero. Last month, police in Camden, New Jersey teamed up with several Good Samaritans to rescue 4 people from a fiery crash. Yeah, all fired. Yeah, we got a trap inside the vehicle. All right, yeah, I got it.
Oh shoot. Dude. Sorry if I was just standing there. I was just watching this crazy scene. Live Rescue. No wonder it's a new show. It's interesting to watch.
Recognize those 4 Good Samaritans to come forward so that they can be commended.
It's a remote.
How does that happen when people just help and then disappear?
It actually happens quite a bit. We have a problem sometimes in law enforcement where witnesses, even on crimes that we want them to stay around, disappear. But on something like this, they should be commended for it. A lot of times they're recognized by the police department or even like the mayor, you know, the city council, something like that. So I hope they do find those guys.
Easy.
We're gonna take a peek. She's obviously not parked any building next to any buildings. So no exposures.
All right. I'm just gonna go through the class, I guess. Oh wait.
So it's not your van.
No, this is our van too, but it's our green van. That's my van too.
So this is the offender.
This is the offender.
Okay. It was parked up there originally. Okay.
And Randy said, "Oh my God, it's spewing something." Yeah, it's gas. Obviously, you can smell it. Okay. So there was a huge puddle up there, and then he came around this way and parked here. And half underneath the van was all soaked.
Well, that's smart to get away from the.
That's what I figured. I said, "Get away."
I don't want you driving it when it's leaking fuel, but at
We were
least
gonna drive. We were gonna try to drive it back to the place, but you know.
we'll take a peek at that, see where it's leaking from. We'll also go look over there and
Yeah,
see.
AAA said once you guys secure it, then they can come in and tow it back to the. We just got from the mechanic.
Okay. How full is the tank?
I don't know how full it was. It's my husband's, so.
Okay. Do you have the keys for it? So she had the, or I guess the husband just got the vehicle.
How many videos do I have? I don't know if I'm at 100 yet.
And somebody
On my main channel, I'm almost at 1,000. I'm at 88. So once I reach 100, maybe I can do like a big goodnight stream or something. I don't even know. Maybe I can do like the best moments of goodnight streams. Like if anything happens. I'm not gonna like watch the whole 10 hours, but I don't know.
Okay, cool. There you go.
Forgot my playlists.
Want to go see a better description of where she's
Homework and
talking about,
projects. What is this?
and see if we can get some password on it.
Oh, I didn't even,
She didn't say anything?
I need to, hold on, before I study, I need to add some videos to the playlist. I'm also going to be eating some snacks. Pringles. I haven't eat those in a while. Cheddar cheese. I can't go wrong with cheddar cheese. They gotta be my favorite.
Unit is racing somewhere. Let's check in.
Let me turn this down. Turn it down so it won't be too loud and it won't distract me. Oh shoot. I like crumbled chip. That's fine. Yes, I just ate it off of the floor, but you know, it's a 5-second rule. I'm also going to be eating some BBQ chips. Now, it's a good thing I'm not making a mess, like if it's a paper thing, because all I'm doing is just looking on the computer for the studying. Yep, it's not like the midterm or the finals, so I'll get into that. I don't know if I'm going to do a studying video of the finals. Probably not, but I don't know. Right now, this is my 51st. Oh, shit. Right now, this is my 51st school video. Yep, I have to check my playlist. I have to update them. Okay, I think that's it. So. Okay, sorry about the long silence, but all right, all right, I gotta go to Google Classroom. Yep. I didn't notice, but Google said that this week is Teachers Appreciation Week. Basically, I pretty much, you know, I think you just like give gifts to your teachers. I did that all of elementary school, and I think 1 or 2 years in middle school, but I did not do any in high school. I don't think we do this in high school because the testing, because I don't think we had, like in middle school, I don't think we had testing during Teacher Appreciation Week, I don't think, but it's usually I guess it's the second, or it starts the second Monday or the first Monday of May. The first full week, because even though we had some days last week in May, so. Alright, so Google Classroom. Yeah, pretty much most of the classes I have is from Google Classroom. Oh shoot.
So Dan, during the break, you were saying she is really lucky that that happened the way it did, because had she gone to the hospital, this is a big deal.
Yeah, those artificial joints are very difficult to get back into place, and I mean, she just stood up and did it all.
We have like 3 different classes. We had tutoring. I guess it's like it helps you to study, I guess, and regular biology. So I'm a pretty much tell you what my final project is about. Let me check my grades, see if they updated it. My math has been updated, so that's good. I don't know about this one. Well, let me tell you what happened today. I should have said this in the last video. Today was testing for US history, and I think some students already took 1/2 of the biology. I didn't take any of the biology today because I thought that will be tomorrow. So we pretty much, like, they told me to go to this room with other students, and we just did nothing. I thought we were gonna do testing, but we didn't. We watched like a movie, but they keep changing it like after 15 to 30 minutes. So I'm like, okay, I'll just play on my phone. Then I got kind of tired. I just slept, and I didn't really sleep. I slept pretty well, but it's not that comfortable because it was the air condition was on pretty high in that room. I didn't really have a jacket because it's May. It's getting hot now, but I felt kind of cold. Oh well. So I pretty much like put my arms in my sleeves, just pretending yeah. Anyway. Yeah, she didn't upgrade the grades because she didn't read she didn't read she didn't look at the final project I had. So, yeah. Yeah, before I have an 81C in math because I didn't do too good on one or two of the assignments. It used to be a 90 and it went down. It went down before it went down a little low, but it went back up a little bit. So I'm alright with that grade. As long as it's not a D or an F, I'm fine.
What's surprising is calls for abdominal pain is one of the most common calls we get with the
Yeah, because I never had a C in that class until now.
or some sort of
English, I need to like upgrade my grades. I just never had the time to do it.
Obviously treat this guy. He's in good hands. Our paramedics there. And I have a quick programming note for you as well. We're going to be here again live on Thursday for a bonus episode of Live Rescue. Same time, 9 p.m. Eastern. This Thursday here on A&E. There's a quick break and we'll be right back. Live Rescue.
I want you to make yourself feel worse.
Freak, I felt weird. I'm still hungry, man. All I've been eating is chips. Anyway. So she didn't... Oops. Oh, look. Here's the directions. I kind of went over this in the other video. It says all essays should be written in essay form. Introductory paragraph, 3 to 4 supporting paragraphs, and conclusion. That's a total to 6 paragraphs, but one assignment I did, I think 5 or 6, and the other assignment, part 2, I did like more than 6. I did like 8. Because I'll tell you why in a minute. It says if you forgot how to write an essay, look it up on Google. Thank you, teacher. Yes, she actually said that on her directions. Essay should be double spaced. I have no idea what that means, but using the okay. Essay should be no longer than 3 pages, but part 2 has a lot of stuff. Just copying and pasting information interesting without identifying where the information is coming from is plagiarism. The essay over 0. But we didn't just copy and paste. We actually took some of that stuff out and we put it in our own words. I said worms. I said we put it in our own worms. I meant to say in our own words. I don't know why that worms came out of my mouth. That sounded weird. Gummy worms or actual worms? Ugh. I like gummy worms, but not real worms. Screw that. If you are handwriting your essays, F that. You know, don't even try to do handwriting essays. Don't even try to do this handwriting. I don't even know why I'm going over this, even though I already did it, but I'm just telling you what the rules were. Part 1 wasn't that much. Part 1 was about genetic disorders, and part 2 was about genetic mutations. Yes, this is pretty much all we've been learning ever since like February. Genetics, like, geez, why are we learning genetics? I know why, but why are we learning it so long? Can we just like go on to something else? That's why I don't like that class. Part 2 is so much. Research and write essay on genetic mutations. In the essay, you should give step 1, the definition of mutation, step 2, describe point mutation and Franish shift. I don't even know how do you pronounce that? Mutation. Describe the 5 types. Step 3. Describe the 5 types of chromosomal mutations. Deletion, duplication, inversion, insertion, and translocation with examples of each. That's a lot for one direction. 4th step, possible causes of mutation. Step 5, advantages and disadvantages of mutation. That's part 2. Let's check out part 1. Part 1 has 4 steps though, but they're very short. Step 1, the effect on a person's genotype with a disorder. That's not much. Step 2, the effect on a person's phenotype with a disorder. Step 3, health issues resulting from the disorder. And 4, treatments for the disorder. So just give examples for like step 4. That's it. That's not a lot. Step 2 is a lot. Yeah, I was kind of upset at that, but whatever. I did it, so I can't, there's no way I can't get a good grade. Or I should say, there's no way I can get a bad grade. That's what I meant. So yeah, I don't want to go over the final project. I don't know, maybe, is that what the test is mostly going to be about? Genetics? I feel like I need to study, but at the same time, I don't really need to study much. I don't even know how long the biology test is. I don't think it should be that bad. I don't know. I'm gonna just look at the PowerPoint. Oh wait, let me go to like tutoring. This video's almost an hour and I didn't really start.
The 2 victims survived, thanks to the help of the officers and the citizens. And Cameron police are now asking anybody who may have recognized those 4 Good Samaritans to come forward so that they can be commended. Instead, just watching out. How does that happen? People just help and then disappear.
It actually happens quite a bit. We have problems sometimes with law enforcement or witnesses, even on crimes that we want to stay around, disappear. But on something like this, they should be commended for. A lot of times they're recognized by the police department or even like the mayor, even the city council, something like that. So I hope they do that.
Well, help out if you can if you recognize them, certainly. But I want to get us out 5
We did like a practice test for like a preview of this, but I don't know if this is. I don't think it's gonna be the exact same one. I did mutations practice test. I think this could be our finals, but I don't know yet. She didn't say we have to do this because it's a practice test.
Take you to something that happened earlier in St. Louis. A woman who took it very personally after someone ran her over.
Alright, I guess that doesn't. Yeah. Alright, whatever. I don't think this counts. Let's go back to the tutoring. Let's see what they have. They don't have much on here. It says practice. Okay, it's loading. Okay, don't load so long. Usually if it loads for a while, it's the internet, but the internet's actually really good. Maybe if I refresh it. Okay, if I refresh it, nothing's happening. Okay. Well, Ryan, if you're watching this, which I don't think you are, but if you are, yeah, something's going on. Something's wrong. Okay, I wonder if I turn off the lights, can you guys still see me? I don't know. I'll check. I'll test it. What does it say? Oh, okay, it loaded. All right. Oops. Yeah, you guys can't see me. Oh, shoot.
I would, if that was me, I would go to the hospital to get checked out. So you're okay with going to the hospital? Yes, the police are gonna come to the hospital, finish their report, and then go do everything they need to do. So you're okay with? Okay. So was she parked when you were? So she was parked there. When you were getting out, and then she started.
I'm gonna wear this because I'm getting a little cold. So, yeah, I use my blankets when I sleep, even if it's a hot day, because we're inside. Okay, what was I saying? Oh yeah, okay, it loaded. It says practice, so shoot. I think, yeah, we were gonna do this, but I didn't. I was so freaking lazy. Alright, I'm just gonna look at everything before I start. DRC Online Tools Practice. Oh, yeah, that's like the name of the test that we do. I know how to use the tools, so I don't think I need to do that. Biology Leap with answers? What? Why did she put answers? Oh, shoot. What the heck? Oh. Oh, wait, never mind. Dude, when did she post this? Dang it. I'm now looking at this now? Fuck. Dude, I'm fucked. Dang it. Dude. How long is this? Is this like the exact same one? Wait a minute. Dude, what the heck? I hope this is not all of it. Because last year, I don't think it was this much. Dude, it's literally... What the heck? I'm just gonna just scroll down and see. Okay, here's the thing. Like, she posted this, like a preview of it, but it's like in paper form. It's like the old school form. So, it's not going to be like that in DRC. But still, is it going to be this much? See, like when it says stop, it says you have come to the end of session 1 of the test. Review your answers from session 1 only, then close your test booklet It's not going to be on a booklet, is it? I don't think so. It's never on a booklet. Not anymore. It used to be. Dude, I'm looking at like a preview of it. It seems a lot. So how the hell can I study this? Look, it says use the information and your knowledge of science. Okay, thank you. There's 3 sessions? See, some of the students, they did like half of it. So is there going to be 4 sessions? Am I going to do all 4 tomorrow? I don't think I can do that. Because here's the thing. Every time I take these big tests, I get extended time, which means I pretty much have all the time I can take to take this test instead of like rushing. I hate when I have to be rushed. See, most of these are just reading, so what the heck? Dude, this can't be everything. Look, there's like, I'm at like question 36 on session 3. And Wednesday and Thursday is math. Unless on Friday I do, I don't know. I don't know what's gonna happen tomorrow. So there's 3 sessions. Dude, holy shoot, dude. This, how the hell can she post 85 pages on this crap? What the hell?
E-Trade's core portfolios is an easy automated way to invest in. We save you time by building, monitoring, and managing a portfolio for you, and provide all hands-on deck support when you need it. Helping you become top dog.
This message is for all Americans that own $10,000 or more in back taxes to the IRS.
Call the number on your screen now, and we will contact the IRS on your behalf, help you end wage garnishments, stop collection calls, remove tax liens, avoid IRS and state tax penalties, and reduce the amount you owe.
I don't know what this answer key has to do with any of this stuff. What the? Practice test library? Oh, I'm not gonna download any of this stuff. No thanks. Session 1. Wait, oops. What if I print all 86 of the pages? No effing thank you. There's 41, so...
So we need to locate the source of the smoldering. They'll bring the line in and they'll put it out wherever it is. So we got here fast, made sure that the fire didn't go up and everything.
And as they respond to that fire, remember that we ride with these departments all week long, and we want to take you to Mesa, Arizona, where earlier paramedics came to the aid of a woman who hit her head on the windshield after her Jeep collided with another vehicle.
Motor vehicle accident, 2 vehicles, just north of the intersection, unknown extent of injuries. PD still en route.
You said 2 vehicles?
2 vehicles, unknown extent of injuries. Alright, we get there, guys. Make sure we're in our safety vests. Traffic. Good job. motor injury to back. No, I think this was driver here, right? Was this guy in the car too? Okay, so 3 cases total. 2 non-injured, and then this one right here. All right. Was she restrained? She said she was. Okay, and she was restrained, Brendan.
Sweetheart, listen to me. Okay, listen to me. What I want you to do is I'm gonna move. We're gonna move for you, okay? Okay, turn your hips. I just want you to stay straight, all right?
We're gonna put some straps on you just to secure. You feeling all right now?
Mary Jo, are you taking any medicine? No. Do you take any medicine? Yes. What do you take?
And we had a 2-car motor vehicle accident. The patient's stable. She was wearing a seatbelt. In those older Jeeps, it's only a lap belt. So she did come forward and it hit her head on the windshield. So we're concerned about a head injury. We're concerned about her back. Since she does have back problems already, we're going to roll her to the hospital to Levy's right across the street.
Let me get you back out to that fire situation in Sacramento.
Nothing eager? We got smoke, you know, we can't go on the phone and stuff like that. That's why I got my phone's down like that. I don't have a phone for that. It's need a phone something. So we got automatic runs this whole.
Yeah, my great thanks to Tice Tiro, Dan Flynn, and Sean Sexler. And we'll see you back here again on Thursday at 9 p.m. for a bonus episode of Live Rescue. Good night, everyone.
Things in here, but we're not finding any source of a fire inside here. This is a large commercial complex. It's a common attic probably above every suite. So they're going to have to pull some of the ceiling tiles, get up in the attic, check for what might be the source of fire.
I'm going to tell you something. Your license not on it? Is that your license card? What's the guy for what?
So I'm looking at this constructive response scoring information. Description: if you get 0, student's response does not correctly identify the information that can be used as evidence to support the explanation or correctly explain how the evidence supports the explanation, or student's response is blank, irrelevant, or too brief to evaluate. Okay, if you get 1 point, student's response identifies information that can be used as evidence to support the explanation, but does not correctly explain how the evidence supports the explanation. Okay, if you get 2 points, which is the highest, student's response correctly identifies the information that can be used as evidence to support the explanation, and correctly explains how the evidence supports the explanation. Okay, I see. I pretty much, okay, there's like a few constructive responses. I don't even know if it's this much, because I look at the preview, it's 39 freaking questions, and when we did the English one, it was a total of, I don't know, like, I'm gonna think, it was 8, then 12. I think it was about 30, but I don't know. I don't think it's gonna be this much. Because when I heard half and half, like did one session and then do the other session. I don't know. See, this is like I'm looking at directions of like the book, not like the computer. I feel like if I study too much, that might not help either, cause yeah.
That's when Lady drove down to a toll booth where he got stuck in traffic. At that point, he attempted to try to concrete wall.
Seriously, why is this so much?
Once he disabled his vehicle, he was access my vehicle. Pulled him out of his vehicle to lay on the ground and was taken into custody. The last time he was turned over to the FBI for further investigation.
Last year it was not this much at all. It shouldn't even be this much. If last year wasn't that much at all. Or maybe they're not going to use all the questions. I have no idea. But shit. Like, this can't be. Like, this is not even DRC. This is like a booklet, so. I'm not going to look at all this crap. I mean, this is tutoring, so. You know, it's like. I don't know. So if you look, it's constructive response. You get like a perfect for 2 points, I guess. I don't even know. This is so much. This is way too much to look at. This is unbelievable. I'm not gonna look at all this, so. I'm gonna do this though. I don't know if this is gonna grade. Is this like extra credit? I don't even know if it is or not. What else is there here? Standardized test taking strategies. What the hell is this? I'm getting a little sleepy, so. How long is this video? Like an hour and a half? Hour and 10 minutes. Test taking tips. The following test taking tips are for. Okay. This is like. Okay, I'm gonna read all this. It's test taking tips. I guess for how to how to I don't know. I guess how do you do it right or something? I haven't even looked at this yet, so. There's 14, I believe. Yep, 14. I don't know it's this much for test-taking strategies. Attempt to answer all of the questions and do not leave any blanks. Okay? I never leave any blanks. There is no penalty for guessing, and students can get partial credit on the open-ended constructive response questions. Number 2, use time wisely. Well, to me that doesn't matter because I get extended time. If you get stuck on a question, make the best guess or place a mark in the test stat item and go back to it after finishing that section of the test. Number 3, get a solid good night's sleep. Okay, I need to do that before the test. Being well rested will sharpen the mind and aid your memory. Eat good breakfast in the morning of the test. Number 4, try to make the morning of the test a pleasant one. Avoid stress. Okay, so just like don't stress about it. Number 5, go to school on time, day of the test. Well, that doesn't really matter because we don't go straight to our testing room. We just go straight to our regular room, then we go and make an announcement. Number 6, listen carefully to the instructions from the teacher and read the directions to each question carefully, especially the constructive response and written questions. Be sure you understand exactly what you have to do for each part of the test. Okay? 7, look for the keywords in the test. Directions and questions such as choose, describe, explain, compare, identify, similar, expect, not, and but. Read each question carefully and think before you answer. Be sure that you understand the question before you start. Compare answer choices. Look at all answer choices and choose the best and most complete answer. Number 9, a word of caution about changing answers. Usually your first choice is correct. If you have to change an answer, be sure to erase completely. Or we don't erase because it's not on paper. What if it is on paper? I'll be kind of pissed. Because especially for the writing. 10, stay focused on the test even if other students finish early. Don't get distracted. Alright, I won't be. 11, annotate. Underlying important words, etc. We can't do that. I think we can do, there's like a tool we can highlight words on the computer. But don't forget to mark your answer. 12, for constructive response and written questions, many students lose points because they do not read the directions carefully for these kinds of questions. Watch out for those questions that have 3 or more parts. Fuck. Read a question in full before you're trying to answer. At least some of the answer choices will be designed to trap students who don't read the question carefully. Also identify the answer first before looking at the answer choices, since some of those choices will be designed to prime you into misinterpreting the question. Don't jump to conclusions. And the last one, eliminate any answers that you know are incorrect, especially on questions where you are having trouble arriving at the answer. Eliminating a few wrong answers can increase the chances of a random guess being correct. Okay. That helped me. Too bad I can't take that with me on the test, because we're not... I forgot to tell you, we can't have our book sacks or our phones, so... yeah. Alright. Shoot, dude. Biology, man. I don't like it. But... you know what? I think it's pretty much like English. You know, English, it's a lot of reading and a few writing. So it's pretty much going to be about this, but maybe I can do good. Who knows? I'm going to try. I really am. Alright, I'm on the regular biology class. I'm going to look at classwork. Okay, got study guide, review, practice test below. Okay, no, this is not it. This is like old. Lesson 1, Lesson 2, Lesson 3, Lesson 4. Additional study material. What? Oh.
Stay in bed, okay? You need to stay in bed for the night. We come back, they're gonna take you, okay? So you're gonna stay in bed for the night. Okay, you don't wanna go to the hospital, stay in bed for me. Yeah, you're going. We still have to get bed.
What the hell? See, there's some text I could read to help me study. Non-Mendelian inheritance. We never study that. Mendel's laws. I think we look at that like once. Human genome, human chromosomes. We look at that one. Genetic linkage. I think we look at that one. Mendelian inheritance. We did not look at that one. Khan Academy DNA. Okay. I might turn off the lights to read this. So I'm gonna turn off the lights.
Next level clean, next level care. There's always a way to make life better. Philips Sonicare.
Twisted tea is the perfect daytime drink, made with real brewed tea and just a touch of
Okay, I know you guys can't see me right now, but that's fine. Because I believe. Can you guys see me now? Oh shit, you guys can't see me now. Freak. How about now? Oh shoot. You guys can see me because I turn up the brightness. How about now? Wait. Okay, you guys can see me a little bit, but it's fine. I'm kind of close to the computer anyway. I'm gonna look at this biology leap 20. Okay. Use the information and your knowledge of science. Thank you for telling me that to answer the question. During an investigation, 2 German shepherds with long hair were each bred with a German shepherd with short hair in a first-generation cross. The offspring from each first-generation cross were then bred to produce a second-generation cross. Which questions could be answered using the results of this investigation? Circle the 3. Okay, well, we're not actually circling, but is the length in the German shepherds a dominant or recessive? That's one of them. This is circle the 3. Okay. How often does the mutation for long hair randomly occur? Okay, that don't matter. How does the protein made? Does it even talk about protein? No. Is long hair in German shepherds an inherited trait? What percentage of offspring? Okay. The model below can explain what facts about photosynthesis. Okay, check all that applies. How energy is stored with carbon. I can't. Dude, how am I supposed to remember all this? See, it's not like. See, math, it's a lot, but some of them we can remember. Yeah, the math videos are going to be longer tomorrow, so It's going to be a lot more. Information about infectious disease. One reason, it's a choose answer. One reason that the number of deaths due to measles is significantly lower than the number of deaths due to. One reason that the number of deaths due to is the introduction of. I don't know. Vitamin A? What? What does vitamin A have to do with this? Fever reducers, vaccines. Okay, I'm not gonna actually submit this. Oops. The large number of cases of tuberculosis. We've never heard of this word. You know what? Oh, here's a reading. Let's try that. Oh shoot, this is a lot. I'm gonna just, like, if, you know what? I think looking at the question first before reading might be a better idea. I'm gonna try it that way. Okay, which claim is supported by evidence from graph 1? So we gotta look at graph 1. I know I'm not showing you guys what I'm looking at, but that's fine. I'm just talking. Graph 1, shell design and habitat. Meadow, hedge, wood. Graph 1 shows the percentages of snails with different shell colors and band patterns found in different habitats. Climate can play a significant role in the survival advantages of each color and banding pattern. Snails with darker color shells tend to be found in living farther north than those with lighter color shells. Scientists have observed that the climate in Central Europe is changing faster than other areas. This impacts not only the local organisms, but migratory species of birds and animals as well. I have no idea what I just read. Snails with yellow shells live in all 3 habitats. I don't like going like up and down. Alright, you know what? I'm probably just gonna go. I can look at this Khan Academy. It might be a video. Okay, here we go. DNA. See, so we can't remember all this crap. Seriously, because we need to remember stuff from math as well. Hopefully the video won't be too loud. Oh. Oh shoot, it's 13 minutes.
Have been around, I could imagine that they've noticed that offspring tend to have traits in common with the parent. For example, someone might have told you, "Hey, you walk kind of like your dad."
I'm watching the
or
video.
Your smile is kind of like your mom, or your eyes are like one of your
Okay, so
uncles.
you guys can't see me, but I'm just gonna watch this video. So I don't know if you guys can hear the voice. I have earbuds, but they're not that great anymore, so I might get new ones. So this is DNA biomolecules. All right, I'm just gonna eat and watch this. I know you guys can't see the video, but maybe you guys can hear it and understand it. All right, here we go.
As long as human beings have been around, I could imagine that they've noticed that offspring tend to have traits in common with the parent. For example, someone might have told you, "Hey, you walk kind of like your dad," or "Your smile is kind of like your mom," or your eyes are like one of your uncles or your grandparents. And so there's always been this notion of inherited traits. But it wasn't until the 1800s that started to be studied in a more scientific way with Gregor Mendel, the father of genetics. But even then, even Mendel, who was starting to understand the mechanisms of, or he was trying to understand how inheritance happened, and you even could start to breed certain types of things, even he didn't know exactly what was the molecular basis for inheritance. And the answer to that question wasn't figured out until fairly recent times, until the mid 20th century, not until the structure of DNA was established by Watson and Crick. And their work was based on the work of many others, especially folks like Rosalind Franklin, who essentially provided the bulk of the data for Watson and Crick's work, Maurice Wilkins, and many, many, many other folks. But it was really the structure of DNA that made people say, "Hey, that looks like the molecule that's storing the information." Just to be clear, DNA wasn't discovered in 1953. DNA was discovered in the mid 1800s. It was this kind of this molecule that was inside of nuclei of cells, and for some time, people said, "Oh, maybe this could be a molecular basis of inheritance." You know, you can imagine what you would need to be a molecular basis of inheritance. It would have to be a molecule or a series of molecules that could contain information, that could be replicated, that could be expressed in some way. But it wasn't until 1953 when this double helix structure of DNA was established that people said, "Hey, this looks like our molecule." So first, let's just talk about the structure here, and then actually we'll talk about where this name DNA, deoxyribonucleic acid, comes from. And then we'll talk a little bit about why the structure lends itself well to something that stores information, that can replicate.
Okay, sorry for pausing. I actually like this better than reading. Because if I read a lot, then I might forget what I'm reading and my eyes might get tired. So I think the video is better. It actually is. For math and science, to me, I think it's better. So it might be for you, it might not be. So yeah. Alright, let's continue.
Its information and that could express its information. We might go in depth on the expression of information in future videos. So this structure right over here, and this is a visual depiction of a DNA molecule, you can view this as kind of a twisted ladder. It has these two, I guess you could say, sides of the ladder that are twisted. That is one side right over there, and then it is another side. There's another side right over here.
Dude, he's
And
like outlining the
those
freaking
two
ladder,
sides, or
whatever
connecting those
that
two
thing
sides of that
is.
twisted ladder, you have these rungs. And these rungs are
There's no
actually
such thing
where
as a
the
twisted ladder, but
information, the
okay.
genetic information, is, I guess you could say, stored in some way. Because these rungs, it's a sequence of different bases. And when I say bases, you're going to say, "Wait, this says acid. Why are you saying bases right over here?" Well, the word deoxyribonucleic acid comes from the fact that this backbone is made up of a combination of sugar and phosphate. And the sugar that makes up the backbone is deoxyribose. So that's essentially the D in the DNA. And then the phosphate group is acidic, and that's where you get the acid part of it. And nucleic is, this was found in nuclei of cells. It is nucleic acid, deoxyribonucleic acid. But it's not. It is actually mildly acidic all in total. But for every acid, it actually also has a base, and that those bases form the rung of the ladders. And actually, each rung is a pair of bases. And as I said, that's where the information is actually stored. Well, what am I talking about? Well, let me talk about the four different bases that make up the rungs of a DNA molecule. So you have adenine. Adenine. And so, for example, this part right over here, this section of that rung might be adenine. Maybe this right over here is adenine. This right over here. Remember, each of these rungs are made up by, it's a pair of bases. That might be adenine. Maybe this is adenine. And I can stop there. Oh, maybe I'll do a little more adenine. Maybe that's adenine right over there. And adenine always pairs with the base thymine. So let me write that down. So adenine pairs with thymine.
Oh, okay, I get
So if
that.
that's an adenine there, then
Okay,
this is going to be a thymine.
I get that.
If this is an adenine, then this is going to be a thymine. Or if I drew the thymine first, well, I'll say, okay, it's going to pair with the adenine. So this is going to be a thymine right over here. This is going to be a thymine. If I were to draw this, this would be a thymine right over here. Now, the other two bases, you have cytosine, which pairs with guanine, or guanine that pairs with cytosine.
Okay, yeah.
So, guanine. We're not going to
I
go into
remember this part.
the molecular structure of these bases just yet. Although
Sorry
these
if you
are
guys
good,
don't know what I'm talking
good
about,
names
but
to know.
I'm pretty
They show
much
up a lot
talking to
in your
myself,
form, kind of
so.
code, your genetic code. Guanine.
Oh.
Guanine pairs with cytosine. Guanine
I
and
just forgot the
cytosine.
name. We'll just say the first letter of the word.
So
A to
actually,
T,
this
G to
is
C.
some cytosine there.
See, I remember that the
Cytosine
most,
right
so.
over here. Maybe this is cytosine. Maybe this is cytosine. Maybe this is cytosine. This is cytosine. And maybe this is cytosine. Then it always pairs with the guanine. We're not gonna. So this is guanine. Then this will be guanine. This is guanine. This is guanine. I actually didn't draw stuff here, but this is guanine. I didn't say what these could be, but these would be made of pairs of. They could be adenine thymine pairs, and it could be adenine on either side or the thymine on either side, and they could be made of guanine cytosine pairs, where the guanine or the cytosine is on that side. Actually, just to make it a little bit more complete, let me just color in the rungs here as best as I can. So those are guanine, so they're going to pair with cytosine, pair with cytosine, pair with cytosine. And when it's drawn this way, you might start to see how this is essentially a code. The order of which the bases I guess the order in which we have the sequence of these bases essentially encode the information that make you you. And you could debate, well, how much of it is nature versus nurture? And when people say nature, it's literally genetic, and that's an ongoing debate. But it does code for things like your hair color. When you see that your smile is similar to your parents, it is because that information, to a large degree, is encoded genetically. It affects a lot of what makes you you, and actually not even just within a species, but also across species. What humans have more genetic material in common with other humans than they do with, say, a plant. But all living creatures, as we know them, have genetic information. This is the basis by which they are passing down their actual traits. Now, you might be saying, well, how much genetic information does a human being have? And the number will either disappoint you, or you might find it mind-boggling. The human genome, and every species has a different number of base pairs, to a large degree correlated with how complex they are, although not always. But the human genome has 6 million. Sorry, not 6 million, 6 billion. 6 million would be disappointing. Even billion might be disappointing. 6 billion base pairs. 6 billion base pairs. And when you have your full complement of chromosomes, and this is in most of the cells in your body, outside of your sex cells, the sperm or the egg cells, this is going to be spread over 46 chromosomes. 46 chromosomes, or I guess you could say 23 pair of chromosomes. So if you divide 6 billion by 46, you get a little over, on average, 100 million. I think it's 100 and something million base pairs per chromosome. And some chromosomes are longer. Actually, some of the longest are over 200 million, and some might be shorter. That's just on average. Now, this number to some of you, might be exciting. You're like, oh, I thought I was a simple creature. I didn't know I was this complex. 6 billion. That's a lot of base pairs. That feels like a lot of information. For others of you, it might not feel so great. You might say, hey, wait, I could store this much information on a modern thumb drive or on a hard disk. I thought I was more unique than that. Of course, we all are special and unique. But, 6 billion base pairs. I thought I was complex. But there's argument for that. Other information. But this is the approximate length, I guess, the approximate size of the actual human genome. And when we talk about chromosomes, and we'll talk about chromosomes in much more depth. Imagine taking this zoomed in thing that you have right over here. And over here, I don't know how many we have, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. We have about 20 base pairs depicted here. Imagine if you had about 200 million of these base pairs, and then you were to take this thing and you were to kind of coil it up into that thing is a chromosome. Is a chromosome. And you're saying, wait, I have that much information in most of the cells of my body? This thing must be incredibly compact. And if you said that, I would say, yes, you are correct. The radius the radius of the DNA molecule is on the order of 1 nanometer. 1 nanometer, which is a billionth of a meter. So you can start to assess kind of the scale of this thing. This is a very dense way to actually store information. But just to have an appreciation of, and you might have seen it when I was coloring in, on why the structure lends itself to being able to replicate the information or even to be able to translate or express the information. Let's think about if you were to take this ladder and you were to just kind of split all the base pairs. So you just have 1/2 of them. So you essentially have half of the ladder. And so if you only have half of the ladder, you're able to construct the other half of the ladder. Let's take an example. Let's say, and I'll just use the first letter to abbreviate for each of these bases. So let's say you have some. So let's say this is this is the sugar phosphate backbone right over here. So this could be one of the sides. Let's say there's some adenine. I should do it in the right color. So you've got some adenine, maybe some adenine right over here. Maybe there's an adenine there. Maybe you have some thymine, maybe thymine right over here. You have some guanine, and then let's say you have some cytosine, and you have some cytosine. So with just half of this ladder, I guess you could say, you're able to construct the other half, and that's actually how DNA replicates. This ladder splits, and then each of those two halves of that ladder are able to construct versions of the other half, or versions of the other half are able to be constructed on top of that half. So how does that happen? Well, it's based on how these bases pair. Adenine always pairs with thymine if we're talking about DNA. So if you have an A there, you're going to have a T on this end, T on this end, T's right all over here, T right over there. If you have a T on that end, you're going to have an A right over there, A. If you have a G, a guanine on this side, you're going to have a cytosine on the other side. Cytosine. And if you have a cytosine, you're going to have a guanine on the other side. And so hopefully that gives you an appreciation of how DNA can replicate itself. And as we'll see also, how this information can be translated to other forms of either related molecules, but eventually to proteins. And just to kind of round out this video, to get a real visual sense of what the DNA molecule looks like, or I guess a different visual depiction from this, I found this animated GIF that, if you haven't fully digested what a double helix looks like, this is it. And you see here, you see your sugar phosphate bases here. You see kind of the sugars and the phosphates alternating along this backbone. And then the rungs of the ladder are these base pairs. So this is one of the bases. That's its corresponding, I guess you could say, partner. And you can see that along all the way up and down this molecule. Very exciting.
Okay, that was a video of the DNA. Yeah, I remember that. I remember that a lot, so hopefully that will be on there. Alright.
Time flies when you're having fun. MLB Network scripts this sits through all the highlights and gets a look ahead always on lightning fast life. It's time well spent. Quick pitch tonight, 1 a.m. Eastern, only on MLB Network.
I'm heading to CMA Fest in Nashville. Everything down to 15. Visit MLB.com/CMA Fest for a chance to win. Maybe you'll have these here too.
Is most impressive pitching performance you've ever attended. Responses to this. 17K hit the favorite into the Yanks with no touch of the dugout. So it might be the All-Star. No one like the 7th field hitter, 91. Come on, Bird was published 2011. Come on, Bird, boy. Not the one. The favorite was against his best
Okay, I think there's another video I can watch. Hopefully it's a video. Yep. Information of transcription and translation. I remember that, but I want to watch this again, if I haven't done that yet. Oh shoot, what the hell happened? Okay, you have to click play like twice, that's weird.
We've already talked about how DNA's structure as this double helix, this twisted ladder, makes it suitable for being the molecular basis of heredity. And what we want to do in this video is get a better appreciation for why it is suitable and the mechanism by which it is the molecular basis for heredity. And we're going to focus on a conceptual level. I'm not going to go into all of the, I guess you could say, biochemical details, really just give you the conceptual idea of what happens. So right over here, this could be a fragment of DNA. I have what I have. This is 8 base pairs depicted. And just to be clear, we talked about this in the introductory video to DNA. DNA is much more than a handful of base pairs. A DNA molecule can be tens of millions of base pairs long. So, for example, this might be a section of a much longer molecule. So, a much longer strand of DNA. And even there, I'm probably not giving justice to it, but this might just be this very, very small section right, we just in a different color, this little section right over here, zoomed in. So once again, it might be part of a molecule that has not 7 or 8 base pairs, but it might have 70 million base pairs. So just like that. So just like that. So let's understand what a molecular basis of heredity would need to do. Well, first of all, it would need to be replicable, or something. We would need to be able to replicate it. As a cell divides, the two new cells would want to have the same genetic material. So how does DNA replicate? And this process is called replication. So let me replication. And we covered this in the introduction video as well, but it's nice to see the different processes next to each other. And replication, well, you can imagine taking splitting these two sides of the ladder. And actually, let's do that. So let me copy and paste. So if I take that side right over there, so let me copy and then paste it. And then there we go. A little bit of it is dropping below the video, but I think that serves the purpose. And then let's copy and paste the other side. So let me select that, and then I copy, and then I paste, and it's just like that. And so you can imagine, if you were to split these I guess you could call them two sides of the ladder, then either side could be used to construct the other side, and then you would have two identical strands of the DNA. And so let's see what that actually looks like. So let me get my pen tool out now. Let me deselect this. Get the pen tool out. It's a new tool I'm using, so let me make sure I'm doing it right. All right, so from this side, from this left side, or at least what we are looking at is the left side, you can then construct another right side based on this information. A always pairs with T, if we're talking about DNA. So adenine pairs with thymine, just like that. Thymine pairs with adenine. Let me do that a little bit neater. Thymine pairs with adenine. Guanine pairs with cytosine. Cytosine pairs with guanine, falling a little bit down here. And just like that, I was able to construct a new right-hand side using that left-hand side. So maybe I'll do the new sugar phosphate backbone in yellow. And we can do the same thing here using the original right-hand side. So using the original right-hand side, once again, the T's pair with the A's, let me do that adenine's color. So we have an adenine and thymine, adenine and thymine, adenine and thymine. Thymine pairs with adenine, so thymine, adenine, thymine, adenine. Guanine pairs with cytosine, guanine, and then cytosine pairs with guanine. So cytosine, just like that. And so you can take half of each of this ladder, and then you could use it to construct the other half. And what you've essentially done is you've replicated the actual DNA. And this is actually a kind of conceptual level of how replication is done before a cell divides and replicates, and then the entire cell duplicates itself. So that's replication. So the next thing you're probably thinking about, okay, well, it's nice to be able to replicate yourself, but that's kind of useless if that information can't be used to define the organism in some way, to express what's actually happening. And so let's think about how the genes in this DNA molecule are actually expressed. So I'll write this as expression. And actually that actually warrants a little bit of a detour, because you hear sometimes the words DNA and chromosome and gene used somewhat interchangeably, and they are clearly related, but it's worth knowing what is what. So when you're talking about DNA, you're talking literally about this molecule here that has this sugar phosphate base, and it has the sequence of base pairs. It's got this double helix structure, and so this whole thing, this could be a DNA molecule. Now when you have a DNA molecule that's packaged together with other molecules and proteins and kind of given a broader structure, then you're talking about a chromosome. And when you're talking about a gene, you're talking about a section of DNA that's used to express a certain trait, or actually used to code for a certain type of protein. So for example, this could be, this whole thing could be a strand of DNA, but this part right over, let's say in orange, I'll do it. This part in orange right over here could be one gene. It might define, it could, this information for one gene, it could define a protein. This section right over here could be used to define another gene. And genes could be anywhere from several thousand base pairs long, all the way up into the millions. And as we'll see, the way that a gene is expressed, the way we get from the information for that section of DNA into a protein, which is really how it's expressed, is through a related molecule to DNA, and that is RNA. And actually, let me write this down. RNA. So RNA stands for ribonucleic acid. Ribonucleic acid. Let me write that down. Ribonucleic acid. And so you might remember that DNA is deoxyribonucleic acid. So the sugar backbone in RNA is a very similar molecule. Well, now it's got its oxy. It's not deoxyribonucleic acid. It's ribonucleic acid. The R, let me make it clear where the RNA comes from. The R is right over there. Then you have the nucleic. That's the N. It's found, it's nucleic, and then it's A, acid. Same reason why we call the DNA nucleic acid. So you have this RNA. So what role does this play as we are trying to express the information in this DNA? Well, the DNA, especially if we're talking about cells with nuclei, the DNA sits there, but that information has to, for the most part, get outside of the nucleus in order to be expressed. And one of the functions that RNA plays is to be that messenger between a certain section of DNA and kind of what goes on outside of the nucleus, so that can be translated into an actual protein. So the step that you go from DNA to mRNA, messenger RNA, is called transcription. Let me write that down. Transcription. And what happens in transcription? Let's go back to looking at one side of this DNA molecule. So let's say you have that right over there. Let me copy and paste it. So there we go. Actually, I didn't want to do that. I wanted the other side. So actually, I think I'm on the wrong. Let me go back here. And so let me copy and then let me paste. There we go. So let's say that you have part of this DNA molecule, or you have one half of it, just like we did when we replicated it. But now we're not just trying to duplicate the DNA molecule. We're actually trying to create a corresponding mRNA molecule, at least for that section at least for that gene. So this might be part of a gene that actually, whoops, let me make sure I'm using the right tool. This might be part of a gene that is, this section of our DNA molecule right over there. And so transcription is a very similar conceptual idea, where we're now going to construct a strand of RNA, and specifically mRNA, because it's going to take that information outside of the nucleus. And so it's very similar, except for when we're talking about RNA, adenine, instead of pairing with thymine, is now going to pair with uracil. So let me write this down. So now you're going to have adenine pairs not with thymine, but uracil. DNA has uracil instead of the thymine, but you're still going to have cytosine and guanine pairing. So for the RNA, and in this case the mRNA, that's going to leave the nucleus, A is going to pair with U for uracil. So uracil, that's the base we're talking about. Let me write it down. Uracil. Thymine is still going to pair with adenine. Just like that. Guanine is going to pair with cytosine. And cytosine is going to pair. Cytosine is going to pair with guanine. And so when you do that, now these two characters can detach, and now you have a single strand of RNA, and in this case messenger RNA, that has all the information on that section of DNA. And so now that thing can leave the nucleus, go attach to a ribosome, and we'll talk more about that in future videos, exactly how that's happened. And then this code can be used to actually code for proteins. Now, how does that happen? And that process is called translation. So translation, which is really taking this base pair sequence and turning it into an amino acid sequence. Proteins are made up of sequences of amino acids. So translation. So let's take our mRNA, or this little section of our mRNA, and actually let me draw it like this. And let's see, it is UAC, so it's going to be UAC, then U, then ACG, okay? And then we have an A, let me make sure I change to the right color. We have an A there, and then we have this UUA, CG, all right, now let me put a C right over there. I'm just taking this and I'm writing it horizontally. I have a C here, not a G, it's a C. And then finally I have a G. And of course it'll keep going on and on and on. And what happens is each sequence of 3, and you have to be very careful where it starts, and so this is in some ways a delicate and surprising, but at the same time surprisingly robust process. Every 3 of these bases code for a specific amino acid. And so 3 bases together, so these bases right over here, these I guess you could say this 3-letter word, or this 3-letter sequence, that's called a codon. And this is going to be the next codon. And we actually haven't drawn the next codon after that because we need 3 bases to get to the next codon. And how many possible codons do you have? Well, you have 1 of 4 bases, and you have them in 3 different places, so you have 4 times 4 times 4 possible codon words, I guess you could say, and 4 times 4 times 4 is 64. So you have 64 possible codons, which is good because you have 20 possible amino acids. So this is overkill and allows codons to be used for other purposes as well. And they also, you might have more than one codon coding for the same amino acid. So you have 64 possible codons. They need to code for 20 amino acids. And so this codon right over here, with the ribosome, and we'll talk more about how that happens, could code for amino acid 1. So let me just write it here. This is amino acid 1. And actually this amino acid is brought to here. They're actually matched together by another type of RNA. This is mRNA we're talking about right over here. This is mRNA. But there's another type of RNA called tRNA that essentially brings these 2 characters together. So the tRNA, and I'm just going to, it's got some structure here. I'm not drawing it completely right, but it's going to match right over here where maybe it has an A, a U, and a G right over here, and on this end, it was attached to this amino acid, and so it matches them together. And then you're going to have another tRNA that might attach to amino acid 2, which I will do in purple, amino acid 2, and that just happens to coincide with, so it can complement right over here, so it attaches in the right place. So it's A, U right over here, this tRNA. And so it'll construct the sequence of amino acids. And as you put these amino acids together, then you are actually constructing a protein. So a protein is essentially a sequence of these amino acids put together. So a sequence of these amino acids put together. These proteins are essentially the molecules that run life for the most part. Obviously, if you eat an animal, it's going to be made up of fat and sugars and proteins, but the proteins are the things that actually do a lot of whether they're enzymes, whether they're structural, the muscle is formed from proteins. These are the things, and I'm just drawing a small segment of them. They could be thousands or more of these amino acids long. And they kind of form these incredibly complex shapes, and they have all of these functions. This is what's kind of doing the work of life, and this, for the most part, and this is kind of how the information for life is stored.
Yeah, that was a long video about replication, transcription, and translation. That should kind of help me quite a bit. So, what the heck?
I've been watching games for years in San Diego, and if you want to judge, and we have all the StatCast data and everything, it's nice to have that data, and they try to project this into it.
She made a rap, or just a rap video about it. No, thank you. I'm not watching that or listening to that.
If this guy is able to, I don't know, just replicate what he's been doing, we're talking pretty much 40, 50 home runs.
I don't know if mutations or genetic disorders are going to be on here. That's why I don't know what to study exactly on this. And it's been almost 2 hours. So I'm gonna go when it hits 2 hours. So I think that's about enough for me today. So yeah, I'm gonna go get something real quick, so I'll be right back. I'm gonna turn on the light.
Life doesn't update you about your credit. So meet Eva, the Capital One assistant that catches things that might look wrong and helps you fix them. What's in your wallet?
This is the couple who wanted to get away, who used Expedia to book the vacation rental that led to the ride, which took them to the place. And they discovered that sometimes a little downtime can lift you right up. Flights, hotels, cars, activities, vacation rentals, Expedia, everything you need to go.
Okay, guys. I'm gonna stop the video when it hits 2 hours. So that was me studying for biology. I'm sorry for being quiet because I had to do a lot of reading and look at all that stuff. So tomorrow might be a lot longer. Will be math. So yeah, thank you guys for watching, and I'm gonna upload this right now. See you guys tomorrow. Peace.